ELISA is an important topic of immunology for DMLT and BMLT students because dengue, thyroid tests, vitamins, and others hormonal tests are done by this technique in the laboratories. Nowadays, ELISA is replaced by CLIA method, chemiluminescence immunoassay method. But ELISA is the basic important topic of theory and practical for MLT students and professionals. Okay, my name is Saurav and you are watching MLT Study. Today's topic of the discussion is about ELISA. What is the full form of ELISA? Enzyme linked immunosorbent assay. I define each of the term one by one. First, enzyme. Enzyme used in ELISA is horseradish peroxidase. HRP enzyme. This is the most common and most widely used enzyme in ELISA. And this is an important MCQ for the MLT students. Other than HRP enzyme, alkaline phosphatase, beta glucosidase, glucose oxidase are there. Linked. Linked means linking substance. Linking between enzyme and immune reagent. Glutaraldehyde is the linking substance, mainly linking substance in ELISA. Next, the term immuno suggests antigen-antibody reaction. So, by this technique, we can detect antigen or antibody present in patient sample. The antigen may be protein, peptide, hormone, bacterial antigen, viral antigen, etc. An antibody may be antibodies against viral infection, antibodies against bacterial infection. The term sorbent means absorb on solid phase support. For ELISA, we used microtiter wells. One ELISA kit contained 96 microtiter wells, and each of the wells composed of polyvinyl chloride or polystyrene. Antigen or antibody are coated or immobilized on this solid phase support. This is called sorbent. And the last, assay. Assay means technique or method. There is 4 types of ELISA method. Number 1, direct. Number 2, indirect. Number 3, sandwich. And number 4, competitive. I describe later the types of individual ELISA. First, we need to know the definition of ELISA. What is ELISA? ELISA is an immunological assay involving antigen-antibody reaction, which is used to determine the presence or the concentration of antigen or antibody in the specimen by the help of an enzyme-linked immune molecule, especially antibody. So, immune molecule may be antigen or may be antibody. In ELISA, the most common is antibody. And this is a qualitative test as well as a quantitative test, because it can detect the presence or absence of the antigen or antibody in the sample, and also can detect which amount of the antigen or antibody present in the sample. So this is as well as qualitative as well as quantitative. Coming to the next point, specimen for ELISA. Serum, cerebrospinal fluid, sputum, urine, stool, semen, etc. Next point, instrument used in ELISA. Centrifuge machine, micropipette, incubator, washer machine, and ELISA reader. Since sample volume or sample load is low in small laboratories, so that there is no need of washer machine, manual washing is done there. Next point, commercial kit components. Number 1, 96 wells microtiter plate made up by polystyrene. Number 2, sample diluent. This is a protein stabilizer and antimicrobial agent. Number 3, enzyme conjugate. This is HRP linked monoclonal antibody. Number 4, wash buffer. Wash buffer is the concentrated phosphate buffer with surfactant. Number 5, substrate, tetramethylbenzidine or TMB substrate. Number 6, TMB diluent contain hydrogen peroxide. Number 7, positive control. This is the recombinant antigen. Number 8, negative control. This is the normal human serum. Number 9, calibrator. Number 10, stop solution containing H2SO4. And number 11, plate sealers. Coming to the next point, basic procedure of ELISA. First, in the microtiter wells, we add patient sample. After adding sample, we add enzyme conjugate. Then incubation is done. After the incubation, washing is done with the help of wash buffer. After washing, add substrate and incubate the microtiter plate at dark place. After incubation, add stop solution and read results spectrophotometrically in ELISA reader within 15 minutes. This is the basic procedure. This procedure is vary from kit to kit literature. The incubation time is important in this procedure. Coming to the next point, types of ELISA. I previously told that there is 4 types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and comparative ELISA. First, direct ELISA. This is the basic ELISA, and this technique is used to detect antigen in the specimen. What is the principle of direct ELISA? In the microtiter wells, first we add patient sample which contain antigen of interest. After adding sample, we add enzyme conjugate. Enzyme conjugate is horseradish peroxidase linked monoclonal antibody. Then incubation is done. In the incubation time, the antigen antibody reaction will occur. If patient sample contain antigen of interest, this will bind with enzyme conjugate. After the incubation, washing is done with the help of the wash buffer. The importance of the washing is all unbound antibodies are washed away. The amount of bound peroxidase is proportional to the concentration of antigen present in the sample. After addition of the substrate, a blue color develops. The intensity of the color is proportional to the concentration of the antigen in the sample. To limit the enzyme substrate reaction, stop solution is added and a yellow color develops, which is read spectrophotometrically by ELISA reader. Coming to the next point, indirect ELISA. This technique is used to detect antibody in the sample. Sensitivity is increases in indirect ELISA because antigen are coated in the microtiter wells. Then we add patient sample containing antibody of interest. Then we add enzyme conjugate. So the antibody interest is trapped between antigen, captured antigen and enzyme conjugate. That is why it increases the sensitivity of the test. Then washing is done. After the washing, add substrate to get the product. The intensity of the color is directly proportional to the concentration of the antibody in the patient sample. Coming to the next point, sandwich ELISA. This is the most common and most widely used ELISA in immunology. This is very sensitive and very specific ELISA, sandwich ELISA. What is the principle of sandwich ELISA? The microvells are coated with specific captured antibodies. Then the samples are added. If it contains antigen specific to this captured antibody, then antigen-antibody binding occurs. Then added enzyme conjugate, so that a sandwich complex is formed where the antigen is trapped or sandwiched between two antibodies. One is captured antibody, another is enzyme conjugate. Then washing is done so that unbound conjugate is washed away with wash buffer. Then add substrate. After adding substrate, a blue color develops. The intensity of the color is proportional to the concentration of the antigen present in the sample. To limit the enzyme substrate reaction, stop solution is added and a yellow color develops, which is read spectrophotometrically by ELISA reader. By sandwich ELISA, we used to detect dengue NS1 antigen, HBsAg antigen, TSH, etc. Coming to the next point, competitive ELISA. Competitive ELISA is also called as inhibition ELISA. There is a competition between two antigens. One antigen present in serum and another is enzyme linked labeled antigen. The microtiter wells are coated with antibody, captured antibody. Then enzyme linked labeled antigen is added. Then add patient serum. There is a competition between two antigens to bind with the captured antibodies. Then washing is done. Then add substrate to get the product. In the competitive ELISA, concentration of antigen in the sample is inversely proportional to the color. If the sample or serum contains more antigen, there is less combination of antibody to labeled antigen, so less color develops. And if the patient sample or serum contain less amount of antigen, there is combination of antibody to labeled antigen increases, so color increases. Example for detection of HIV antibodies. This is the calibration curve between sandwich and competitive ELISA. Coming to the next point, diagnostic applications or uses of ELISA. HIV detection, torch panel, dengue NS1, dengue antibodies, IgG, IgM, HBsAg detection, HCV detection, Mycobacterium tuberculosis detection, differential hormonal assay in the laboratory. Next point, advantages of ELISA. This is very sensitive and specific immunoassay. It has minimum detection limit of 0.06 microgram per ml. Reagents have long shelf life. This is very cheap method. Equipments are not expensive. Does not require radioactive material, so no radiation hazard. Does not require special license to handle reagent. If you are a MLT student or medical student and visit the channel first time, please subscribe the channel and press the bell icon to get the notification for the latest video.